This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 267, recorded on January 10th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Howdy, Vincent. How are you?
Well, I'm fine, and welcome back.
Thank you. Good to be back.
From the West Coast. You were there for 2 weeks?
I was there for 2 weeks. Almost 2 weeks. Close enough.
Is that enough?
No.
I mean, your kids and stuff, you know, I'm sure you can't get enough of them, but...
No, that's right. No, I think every time we go out to this joint, this is, you know, basically a vacation house in my wife's family.
Yeah.
We talk about how we will probably, in my retirement, spend a lot of time out there. It's real nice.
Yeah, you're going to split your year between Gainesville, Oregon, and Texas, right?
And Massachusetts and Seattle and...
Oh, right.
Yeah, we're going to make a big circular trip.
You're just going to be traveling constantly.
Continuous circular trip amongst all of those locations, with Oregon thrown in there as a West Coast stopping point, and Florida as well. Stop at all the national parks in between. Yeah, we're just going to keep driving.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
You thawed out?
Yeah, it's 36 right now, which
It's
is
ridiculous.
2 Celsius. Yeah, it is crazy.
It was so cold this week, right?
There was one day, I have never done this before, I worked at home just because of the weather. They were saying it was minus 40 with wind chill and life-threatening wind chill, and I could work at home, so I did. It was before classes started.
Yeah, we had this week minus 17 C. And, you know, after last week's episode, we were talking about minus 17. And someone wrote at the website, twiv.tv, "Are you kidding? When it's minus 17, I wear a T-shirt. It's minus 40 now."
Yeah.
It's always colder. Here it's 2 degrees at the moment and terribly cloudy. It started snowing this morning, warmed up. Now it's raining. The Hudson River is very interesting. It's full of what Dixon calls pancake ice. These bits of ice pieces coming down. It comes down from the north part of the Hudson when the tide goes out, and then it goes back up again when the tide comes in. It's very interesting.
Well, it was miserable here for 2 days. It actually hit freezing.
Yeah, I saw a picture of Grant McFadden's on Facebook of a frozen fountain.
Yes. So that's about as bad as it gets here.
You probably feel insulted when it gets frozen, right?
Oh, yeah. Absolutely. That's the proper description. It is now, by the way, 76, which is 24 Celsius and cloudy. But at least the temperatures
Unless
returned.
anybody be concerned, both you and Grant have paid your dues in the cold weather.
Oh, yeah. I'm getting to that. I got a pic that addresses this.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
What's that thing called? The snow woe?
The Wovle.
The Wovle.
The snow wolf or the Wovle. I guess snow wolf sounds better. Wovle sounds kind of...
I got to look this up. S-N-O-W what?
Wolf.
It's great.
I was listening to that podcast this morning. So, there it is. My goodness.
Isn't that cool?
Yeah. Wow.
Yeah, it's a very cool thing. I like it.
Of course, now we got snow this morning, and now we have fog because, of course, the temperature is skyrocketing up, and then we're going to get rain, which is going to flood all the drains that are clogged with the snow now. And
yeah, exactly.
Yeah. So
do you feel silly wielding this thing? It looks like a pretty silly device.
Well, feeling silly hasn't really stopped me from much in life, but
I was going to say.
I read all these very, very positive reviews of the thing, and I said, okay, I'm going to give this a try, because my snowblower is out of commission. I had to take it apart because I hit a stick with it, and it ate up the impeller, which apparently you can't order that part anymore for that particular snowblower. So anyway, I got the Wovle, and cleared cleared several inches of snow off my driveway in the same amount of time that it takes me with the snowblower. So I'm totally sold on it.
Wow.
And no back pain.
Me.
Mine is on the way.
No, you
I got
bought
the
one,
shipping notice. Oh, yeah.
Yeah, you do feel it in your shoulders the next day from launching
Okay.
big scoop loads of snow, but not in your back.
That's probably good.
Yeah. Yeah, it's a good workout.
Well, after we recorded last Friday, I went out with my 3 offspring, and which is unheard of that they all helped me shovel snow, and we cleared the driveway. And of course, Monday everything melted anyway.
Right.
Monday was weird
not
because
ours.
it got warm. And Monday morning, I think I walked out, and there was this wave of hot air. There was fog everywhere because there was still snow on the ground. It was amazing.
Oh, we had incredibly dense fog here
amazing,
on Monday.
amazing stuff. Weird weather.
It's incredible that listeners put up with this stuff, you know?
Yes, This Week in Virology is the show, by the way.
This week in what is that? This week's
week in
weather.
The weather and
Weather in
virology.
Virology,
It's
yes.
TWiV. Well, some people don't like it, and they have told
Tough.
us. All right, we have a couple of follow-ups. First, I have an announcement to make. The American Society for Microbiology is the communications committee, of which I am chair, is planning its communications activities for the next few years. We're interviewing a number of people in different parts of microbiology to find out what they think we should be doing. And we would like to have a few young scientists participate. You should be an ASM member, you should be a grad student or a postdoc, and you should participate in some kind of public outreach activity. And if you're interested in being interviewed, this is being done by a consulting firm on the phone. Please email us an answer to the question, what should be the highest priority goal for ASM's outreach to public audiences and why? Just a few sentences so we can get an idea of where you, if you're interested in this. And you need to do this quickly because we need the answers by January 24th. All right, next from Carol.
So,
yes?
Well, just a question on that. So is there a link, or you want them to email you at TWiV?
TWiV at TWiV.tv. Thank you, yeah.
Okay.
Carol writes, Vince, Kathy, Allen et al. I just saw Emily and the Oats great dengue virus on the podcast. That was Emily's knitted dengue virus pillow. What's really funny is that I also figured out how to knit a virus this past year. What are the odds? In my case, I went for picornavirus, and I made an icosahedron frame out of styrofoam. Oh, interesting. I'm including the link to my Ravelry site where I have the free pattern for any like-minded knitters. So she sends a link to this, and if you click on the little thumbnail, it gets bigger. It is beautiful.
It is.
And it's perfect for today because we have a structure paper. Mm-hmm. You can see the 5, 3, and 2-fold axes of symmetry.
Yep. Mm-hmm. We have a structure paper about a closely related virus.
That's right.
Well, actually, yeah.
Yeah, picornavirus.
Coronavirus.
Ravelry is a free social networking service beta launched in 2007. It functions as an organizational tool for a variety of fiber arts, including knitting, crocheting, spinning, and weaving. Who would have thought?
I guess you can download her pattern, I guess. Mm-hmm. I think fiber art I've never heard before. I really like that. Fiber arts.
Oh, yeah. There's a whole realm of things that you can find out about.
Are you a fiber artist, Kathy?
I've done some quilting, but not really art. But I have some books of fiber art quilts, and
it's cool.
It's amazing stuff.
Wow, so you really nailed this structure.
It's nice. It's perfect. Yeah, it's good. Love the podcast. I recommend it to my students all the time. Carol is a professor at Hofstra University out on Long Island here. Nice. Another episode image.
Great.
Thank you, Carol. Anybody else? Send them in.
Yeah.
I'm sure there must be more viral fiber artists. Carol, now there was a post at TWiV.tv on episode 266 from last time.
This is a different Carol with a K.
Right.
Yeah, that's right. Now, we had talked about a paper showing that HIV-infected CD4 cells are killed by pyroptosis. And Carol wrote, guys, don't you think it's a little weird that a guy who lives 30 years with HIV and is still alive, and he is the example of how HIV kills CD4 cells in vivo? Why would you use an example that contradicts your data? The elite controllers are a small percentage among people who have HIV. They present this process of cell death as fast and efficient, and then they show a guy who presumably dealt with that for 30 years. For me, it means that you can have low numbers of CD4 and live normally without taking any drugs. So I looked back at the paper, and it turns out that the two individuals they used were actually not elite controllers. They have high viral loads and low CD4 counts. Mm-hmm. So I think we were wrong in calling them elite controllers.
But they are people who've had this virus without drug treatment for since the 80s.
Yeah,
right?
Absolutely.
So,
but
they're elite something.
Well, the definition of an elite controller is viral RNA, but they typically have almost normal CD4 counts.
Right.
And if that were the case, then I would agree that how can you study cell killing in someone who's got normal levels of CD4? But these individuals don't, and I don't know why they're still around, to be honest. Now,
maybe they live in bubbles.
I would not use them as an example for not taking drugs if you have AIDS.
No, we know we have ample data that if you go without the drugs, you do die.
Yeah. So anyway, there was
Usually.
a little discussion over there, and thank you, Carol, for posting that. All right, that's it. No more follow-up. Moving into our papers. The first one, actually, Kathy, came to you via Stefan Taub, right? Yes.
I don't remember.
Yeah,
how
in fact,
this came.
yeah,
Yes.
you sent me an email, and it was forwarded from
right.
Stefan. Yeah.
Yes, that's right. Stefan sent it. Yeah.
And this is a paper in PLOS Pathogens. TMPRSS2 is essential for influenza H1N1 virus pathogenesis in mice by Hatsuyer, Bertram, Menert, Bagat, Nelson, Polman, and Shugart.
Who names a gene without any vowels?
It's a Web 2.0, man.
Jeez.
Yeah, no vowels. That's the latest thing in the web. Well, the latest. It's been years now, but
well, I guess if they wanted really to have the latest, it would be iTempers. Right.
That's right. A little lowercase I. This is a very cool paper. It is a short and sweet paper, but not short in work. There's a lot of work here, and this revolves around the known fact that influenza virus hemagglutinin needs to be proteolytically cleaved in order for the virus to be infectious. And the protease that does this in an animal, you can in cell culture you can add trypsin and it will cleave the protease and you can get your viruses to replicate. So as the viruses come out of cells, they have to be cleaved in order for them to be to infect another cell. But in people, it's not clear what the protease actually is, or in animals. And when I lecture to medical students and other types of students, I always talk about this tryptase, or this protease called Clara. There are cells in the epithelium of the respiratory tract called Clara cells, and they produce a protease called Clara, which has been thought to be a possible candidate for this protease. But there's evidence that this protease called TMPRSS2, which stands for type 2 transmembrane serine protease. This is a membrane-associated protease. So it's on the outside of the plasma membrane?
Well, it is a type 2 transmembrane serine protease.
Yeah.
But then it's one of those of that class, and then its name is some abominably long thing that abbreviates to TMPRSS2, which is how it ended up with that.
I think that's type 2 membrane. Oh, I see. TTSP is type 2 trans, and TMPRSS2 is, we don't even know what that's coming from.
Yeah, I'm just looking. I looked for the name earlier.
So I have, while we're looking for this, I have both a comment and a question about this stuff. First of all, this notion that you have to cleave the hemagglutinin to activate it. I realize, Jane, that this is being pretty anthropomorphic here, and it's teleological, and et cetera, but I'm sorry, that's the way my head works. I always think of, so the hemagglutinin causes membrane fusion. It's part of the fusion process for a virus infecting a cell, and it gets synthesized and assembled in the Golgi and processed through the Golgi to the surface of a cell during the maturation of the virus. So as a fusion protein, this is a dangerous protein. You don't want this thing to go off prematurely, or you're going to fuse all the membranes in the cell and that kind of stuff. So logically, it has always made sense to me that you want some sort of safety mechanism to make sure that this thing doesn't go off prematurely. You want to wait until you've made or almost made virus and then sort of cock the gun. And this, to me, is a way of doing that. You do the cleavage at the last minute, and now this thing, you're safe, and this thing is ready to go. Fair enough?
Fair enough.
Yes.
So I think there's been a question as to whether this is the proteolysis is going to, where during the life cycle this is going to happen, whether the protease that's going to do it is going to be an intracellular or an extracellular enzyme. So where do we stand on that?
So the Clara is a secreted protease. So the models have been, the virus is produced, it buds from the plasma membrane or the apical domain of the cell, and then it's cleaved. But as they say in the introduction, there's little evidence that secreted proteases are actually involved. So,
but now you're saying that this thing is maybe a membrane-bound protein?
Well, it says membrane-associated. It could be that...
So the TMPRSS2 is transmembrane protease, comma, serine 2.
Yeah.
So at least they didn't put the comma in the abbreviation. But it is a transmembrane molecule.
So the type 2 means the N-terminus is embedded in the membrane, right?
So that would make sense to me if it were something that could act in particular, I don't know, but in particular at the plasma membrane, like sort of during the budding process or something like that, as a last step in the maturation of the virus.
So that this protease apparently previously has been shown to be able to cleave influenza virus in cell culture. Okay.
Do you have to add proteases to a plaque assay?
Yes. So when we do flu plaque assays, we have, unless you use a strain, there are a few strains. One of them is called WSN. You don't have to add trypsin, but all other strains you add trypsin to the overlay. Yes. Otherwise, you don't get plaques.
So I just gave this lecture today, and I've been giving it for 20 years. That has as part of it, I've talked about quantitative assays of viruses, and there's this table that came out of, I don't know, one of the older, like Delbecco or somebody, microbiology books some time ago that compares all the different quantitative assays on a stock of flu virus. And one is an egg infectious dose assay and another is a plaque assay. And you get out only 10% the infectivity in a plaque assay as the egg infectious dose assay. And I've always wondered if in that particular example the reason was this requirement for proteolyzing the hemagglutinin, or if there's other things going on there.
Well, you add protease, otherwise you can't get plaques at all.
You don't get plaques at all. Okay.
No. So it must be something else, right?
Okay. Good enough.
All right, so in right now, if you do a flu plaque assay, you add trypsin, you buy what's called TPCK trypsin, and you put 2 micrograms per ml in the overlay, and you get nice plaques. So the question is, what's the protease in us? That would obviously be important to know. And by the way, most human strains, the idea is they're limited to the respiratory tract because that's where the right protease is that has to cleave them. And they have a HA, which just has one basic amino acid by the protease cleavage site, and their cleavage is restricted to the respiratory tract. Now, some avian influenza virus strains have multiple basic amino acids around the protease cleavage site, and these can be cleaved by what are known as furins. These are proteases that are found in all tissues. And so these viruses could replicate in theory in many other places. And of course, in birds, they replicate in the GI tract, among other places. So these proteases are thought to be really important for tropism of influenza virus. And so knowing which is the protease might be therapeutically valuable.
Yeah.
So this study, they look at this tempress. Can we call it tempress?
Yeah,
Sure.
good idea.
And they take mice which lack the gene encoding tempress 2, and they ask what happens when you infect them with influenza virus. So these mice have deletion of the gene, they're fine. They have no problems. They can listen to opera and understand it. No, I don't know about that. I don't know about that. Well, Harold Varmus said that
Where
once.
did that come from?
So he said, there's no phenotype in these mice that lack this gene, but we never took them to the opera, so we don't know if there's some other issue.
Yeah, the metaphor was, until you've taken your mice to the opera to see if they can distinguish Wagner from Mozart, then you haven't really characterized their phenotype.
Jeez, I don't know if I could even do that.
Yeah, exactly.
Wagner puts you to sleep.
Oh, okay.
At least it did me. I went to see I went to see some 8-hour Wagner thing in Vienna once with Peter Sarno and Carla Kierkegaard. I could not stay awake. And
Did they go to sleep?
Yeah, no, everybody around me, all the Austrians were awake, and I felt so conspicuous because my head, how your head keeps going down when you fall asleep. I could not take it. There were 4 intermissions.
Did you have a refreshing sleep?
No, because I felt guilty.
It's great. It's a selection for that particular phenotype.
Oh man!
You go in and pick out all the sleepers.
Anyway, so I have no knockouts, I guess. Maybe I do. Anyway, these mice, they infect them with PR8. Now, PR8, if you remember from previous TWiVs, is a strain of H1N1 influenza virus that can infect mice. And when you infect mice, they exhibit weight loss, and half of them die, and the virus replicates in the lung. But when they infect the tempress knockout mice, they don't lose weight. And they show no signs of disease. And they also do a measurement of blood oxygen, which is a measurement of lung function, and this drops in wild-type mice and not so much in the tempress knockout. So that's pretty neat. Pretty neat result. And similar onset of infection in both wild-type and tempress mice, but the virus doesn't seem to spread in the tempress knockout mice as it does in wild-type mice. So this is totally consistent with, you get an initial round of infection because the virus you're infecting them with, of course, has cleaved HA. You've grown it in a way so that it's cleaved, so that it can infect the cells, but it doesn't spread. They also measure infectious particles made, and it's much lower in the tempress knockout, which is consistent with this protease being important.
And they show that the HA is indeed not cleaved.
Right. They pull virus out of the lungs of these mice, and they look at the HA on a protein gel, and indeed it's not cleaved in tempress deficient mice. This is an important experiment because it could be some other effect, right?
Mm-hmm.
Right.
They also tried an H3N2 virus, which is mouse adapted. So it has, other strains don't grow in mice unless they're mouse adapted. There is an effect of the tempress knockout, but not as much as with H1N1. And they speculate there may be other proteases that cleave H3N2 in addition to tempress. It's never black and white. To assume that all strains of flu would be cleaved by the same protease is probably simplistic, right?
Mm-hmm. When they showed this figure and they said that there's not as much of an effect with the H3 virus, I looked at the figure just cursorily to start with, and I said, well, looks fine to me. But then they did 2 different doses, and I looked at it in more detail and realized that where they're seeing an effect, they're infecting with 10 PFU per mouse.
Yes.
Okay, whereas in the H1N1 virus, they're infecting with 10 to the 5th PFU per mouse.
Yeah.
And if they just jack up the dose to, 100 PFU per mouse or something, 10 to the 3rd, 1,000 PFU per mouse, then they don't see as much of a difference.
Right.
So there is a significant difference.
Yeah.
The next experiment is really cool. They use a H7N7 virus, which has a multi-basic HA cleavage site, as we talked about before.
Now, is this the virus in seals? It's A seal Massachusetts 180 H7N7. And I was wondering, is that the one we talked about back on TWiV? I forget the number.
Yeah, it certainly came from that seal outbreak in 1980. Yeah, and so whether that was the same as the one we talked about, I don't know. But these can be cleaved by other proteases, and as you would expect, the mortality and weight loss in the knockout mice, the temperus knockout mice, is not as much as with the other strains. So again, because there are other proteases involved. So that's another nice control there, right?
Yeah, because it's mechanistically consistent with what they think is going on.
And then the last experiment they do is to actually express these temperuses in cell culture, and they show that they can actually cleave HA proteins of a variety of influenza viruses. And they're looking at murine temperus in cell cultures. And so that just shows that in cells you can express these proteins and they will cleave. They're expressing them in a human cell line, I think, and they will cleave influenza viruses. So I guess you could make a cell line expressing temperus and grow your viruses in it.
Bingo.
If you don't want
to do plaque assays. If you want to do plaque assays.
That's right. So there you go. Yeah.
That last experiment was the one that was the supplemental figure.
Mm-hmm. Yeah.
Yeah. As soon as I got to that, I thought, wow, this should be in the main part of the paper. To me, anyway. I thought it was a key point.
Yeah. I never understand why some things are relegated to supplemental. Anyway,
especially in an open access online journal.
Right. I like so basically this could be, who knows if this temperus is doing the same thing in people, of course, right? Because as we know, ferrets are not humans and neither are mice.
Right.
And their last sentence of the paper, it should, however, be noted that our findings in the mouse model system require validation in humans. How would you do that?
Well, you need a temperus knockout human.
First, you find people who fall asleep during Wagner.
Yeah, there you go. Vincent.
It's a good episode title. People who fall asleep during Wagner. I mean, I was thinking, how do you... I mean, you can't do knockouts. You can't do infections. What do you do in people? I mean...
Well, I wonder if there are any mutant alleles of temperus 2 in the human population.
You could look for that. But, you
what
know...
are you going to do with them?
These are... You could take a tracheal explant culture, right? So there are lots of these cultures now that you get little snips of people's sinuses, you put them in culture and you can infect them. And I guess you could see if temperus is expressed in them, but can you knock things down in these cultures? I don't know.
I think before enrolling any patients, I would want to see this in at least another animal system.
The thing you could do it in ferrets, but there are no knockouts or knockdowns in ferrets because they don't do that with those animals, you know.
Right, but you'd at least have a more manipulable system. You might be able to, target a monoclonal against the receptor or something.
Maybe you could treat with an antibody and see if it inhibits it. Yeah.
Yeah.
So the validation in people is a tough one, I think.
Mm-hmm.
Yeah.
Well, let's say you did say you found that temperus is it in humans, then I guess people could design protease inhibitors, right?
Yeah, that's one of their selling points in this.
Yeah, maybe. Really surgically targeting just one
hard.
protein that's part of a whole family of related proteases is going to be a nifty trick. But that might actually be good. I was reading this and they're talking about how you might, by specifically targeting it, you might get fewer side effects. But if you had a little bit of crossover between a few of these different proteases, you might protect against a broader spectrum of viruses.
Yeah, they do note that quite a few other viruses are activated by TMPRSS2, metapneumovirus, MERS coronavirus, SARS coronavirus.
Well, I was even thinking just within the influenza.
Yeah.
Because they've got some flus that are and some flus that aren't, and maybe you want to hit some of Temperus's brethren to have more broad-ranging anti-flu activity.
I think from a basic virology viewpoint, this is interesting because I'd like to know, where this protease is in the epithelium and how it actually cleaves, how it acts, at what point, during budding, after budding.
Yeah.
Those are interesting questions. It doesn't always have to be about drugs, right? Right, Rich?
Mm-hmm. Right.
Yeah, and in fact, all these problems that you could run into using this particular approach to develop a drug, this may not be the approach to use at all. But
right.
getting this kind of detailed mechanistic insight is where, that's where we got the protease inhibitors in the first place.
Right.
So go a step further and figure out exactly which proteases and how they're cleaving, and maybe you can do some entirely different strategy.
Well, for me, I'm going to now revise my lectures, and because I'm always showing this picture of a cell secreting a protease, and apparently it's not the case.
Or may not be the case.
May not be
the case. We will hear more, I'm sure.
Yeah. Well, I always like revising lectures. It's cool. Mm-hmm. All right. The second paper is also in PLOS Pathogens. It was sent to me by Steve Goff. Sent me a bunch of papers in an email the other day, and he said, cool entry stuff. And this one stuck out. It's called Viral Uncoding is Directional. Exit of the genomic RNA in a common cold virus starts with a poly-A tail at the 3' end. And the authors are Harunjanan, Kumar Sedivi, Subirats, Kowalski, Kohler, and Blas. Dieter Blas is the PI. I've known Dieter for years, and in fact, he is at the University of Vienna. Speaking of an opera.
Falling asleep at the opera.
By the way, I don't think we did the authors on the previous paper.
I did.
Did we? You did?
I didn't say where they were from, though.
Was I asleep?
They were also from Germany, I believe, right?
Right. One's from also Tennessee Health Science Center in Memphis and Seattle. So, yeah.
But most of them are in
Braunschweig.
Okay.
Yeah.
All right. Sorry.
So this paper deals with, as you can tell from the title, how picornaviruses release their genomes in the cell. Now, the rhinoviruses are taken up by endocytosis. So rhinoviruses are classified as enteroviruses within the picornavirus family, along with polio. And all those entros seem to be taken up by endocytosis. Many of them don't require acidification in the endosome, in other words, low pH, to release the genome. So polio, apparently interaction with the receptor is enough to uncoat the particle and release the RNA.
Single-stranded positive sense RNA. It is a messenger RNA molecule, basically.
That's right.
And in a naked capsid. So you take up this naked capsid, this nut, and uncoat it somehow and release single-stranded RNA that's going to be directly translated.
Right.
This strikes my first reaction to this paper is that this is backwards.
Yes.
This is not the way I would do it. But
well, it's a good thing you weren't doing it, right?
Yeah, that's right.
Well,
yes.
But Rich, it may have to do with
the
how
encapsulation.
it's packaged. Yeah.
Yes, exactly. We can get to that.
I mean, so you're thinking the 5' end should come out first so it can be translated, right? Yeah.
Yeah.
Yeah. Well, anyway, so polio requires just to interact with receptor, and that's enough to pop the RNA out. But these rhinos, Rhino 2, they're called minor group receptor rhinos because they interact with low-density lipoprotein receptor as opposed to most of the rhino serotypes that interact with ICAM1. These require not only endocytosis but low pH. And how the RNA gets out has been something of an open question for many years. Many people have worked on it. Dieter Blas here on rhinos, Jim Hogle using polioviruses, and many other. And Alan Dove also worked on it.
Of much lesser note than Dieter Blas.
Oh, no, you're a titan in the field, Alan. Because you have this nice proteinaceous shell, which is built with icosahedral principles, and how do you get the RNA out? We had a membrane. If it's flu, it's easy to imagine fusion, but these capsids, how do you get it out? So a lot of work has gone into understanding this, and apparently as uncoding proceeds, channels open up in these virions, and Blas has done a lot of nice structural work on these channels, and there are a couple of them that form on the virion as it uncoats, again in response to low pH. And as he says in the intro, the largest ones are at the twofold axes of symmetry. Here's a good time to look at your knitted picornavirus because that will show you what a twofold axis of symmetry is quite clearly. So, Carol, you're providing an essential service here. Now, Carol has made blue VP1, which is correct, yellow VP2, and red VP3. I'm going to use Carol's image in the episode, so you can go look at it at TWiV.tv. The 5-fold axis is made of 5 copies of the blue knitting material. The 3-fold axis is beautifully visible in this image because it's the 3, I don't know, is it cyan or magenta? Magenta, right? Mm-hmm. And they're coming together like a propeller.
Yeah.
Then the 2-fold axis is where 2 yellow subunits come together. You can see those very nicely. If you go from one 3-fold to another, that's the 2-fold axis along those 2 yellow subunits. Is that clear to everyone? Mm-hmm. So that's the 2-fold axis, and
right,
that's
it's the crease between 2.
Yeah, it's the crease before the goal, right?
Right.
Yep.
So according to the studies of Dieter, the largest channels are at the 2-fold axis, and then there's smaller ones at the 3-fold axis and also near the 5-fold axis as well. And he believes that the 2-fold axis is where the RNA comes out, and that's been suggested by some studies with poliovirus. All right. So the idea is that inside the capsid, the RNA is probably all folded up and it's got a lot of double-stranded regions, and then somehow it's got to get out through this pore at the 2-fold axis maybe, and probably only a single strand can get through because it's not big enough to take a double strand of DNA, of RNA. And he says something that's really interesting several times. He said in all the textbooks, it shows the 5' end coming out first. And I must admit, I am guilty. We are guilty. Because in our textbook, we have, in fact, the 5'.
Because that's the way you would do it.
That's right. That's the logical way to do it.
I don't know.
Well, you had a 50% chance of being right.
Yeah. When we made the figures, I was basically reproducing figures from people who work in the field. I mean, we never did experiments to look at which end comes out first, but other people have, and that's how they drew it. In fact, for the picornas, the 5' end has VPG on it, that little protein. So we show it coming out with that little ball at the 5' end. But that's what this paper is all about, to figure out how it works. And one of the main techniques they use is called fluorescence correlation spectroscopy. Now, I understand that Rich Condit is really into this.
Well, I can give it a whack.
Give it a whack.
I've been thinking about this for a couple of days. So, what's involved here is a setup that uses a modification of a confocal microscope, which really allows you to, through the magic of lasers, to take an optical section through anything. In this particular case, it's just fluid, a solution of something, and to excite fluorescent molecules with the laser and then read the fluorescence out the other end. And in this particular case, what the magic of that, not only do you get an image if you want it, but it allows you to define a very, very small volume. Okay? And you can look at also very small concentrations of things. And so what this does is to take a solution of fluorescently labeled molecules and record the fluorescence intensity over a period of time. And if you just look at the raw data reading out from that, what you see is just sort of a bunch of noise, okay, with fluorescence intensity fluctuating over time because of, I guess, Brownian motion, you'd call it, or thermal motion in the solution of molecules. Things are moving around.
It's like looking at snow in your headlights.
Yeah. So very good. So stuff moves in and out of this volume that you're looking at. So at some point there'll be nothing in it and you get very little fluorescence. Another point, there'll be a whole bunch of molecules in there and you'll see more fluorescence. So without any other signal processing, you just see a bunch of noise. But then they do a thing with this called autocorrelation, which, as I understand it, and correct me if I've got this wrong, is basically comparing the signal with itself. And the way I've thought about it is that if you start off with a given point and you have a given signal, think about it that sometime later, if everything was frozen, in that sample and nothing moved at all, the signal would be the same. And so if you define the signal at time x, signal at time 0 being the same, if you define that as 1, you'd get a straight line with respect to time, nothing changing over time. If, however, a little time later, molecules move around and the signal changes so that it bears no relationship to what you saw initially compared to itself, you'd get a 0. All right. So what happens is that with time, the signal changes and the autocorrelation function decays from 1 down to where it's the initial comparison is exactly the same to sometime later where it doesn't look the same at all, and that's 0. And
that indicates how fast the snow is moving through the headlights.
Exactly. So with respect to time, the normalized autocorrelation function decays from 1 down to 0. Now, if the molecules are really small, they move really fast, and so that decay happens quickly. If they're really big, they don't move as fast, and so that decay happens more slowly. So on this autocorrelation function graph, large molecules are moved to the right, small molecules are moved to the left. So out of this, you can see not only the concentration of molecules in this volume, but you can get some idea of how big they are by how fast they move. Unbelievable.
Great.
Yep. And I mean, this is yet another technique that biologists have for separating big things from little things. But the importance with this is you can look at really, really tiny volumes even inside a living cell.
Right.
So
we're going to see more and more of this.
We're going to see a lot of this in the future. So you can separate these sizes of molecules on a gel, but you have to kill the cell and lyse it and prep it and do all that sort of thing. Here you can actually see in real time over a period of milliseconds. You can measure the diffusion of these molecules in a teeny volume.
Right. So
you can fluorescently label more than one molecule and ask whether they're associated with each other, in which case they would have the same autocorrelation function.
Right. And that's what they do here. They say, so they want to they make oligonucleotides, short RNAs, short nucleic acids, complementary to the 5 or the 3' end of the viral RNA. And they're fluorescently labeled, and they say, is this associating with one or the other end? So they test it out first by just taking RNA and the 2 oligonucleotides, and they show they get good correlation of the oligo with the 5 or the 3' end. If you digest the RNA with RNase, there's no correlation, it's fluorescence correlation anymore. So the system works.
Right.
The oligos. Go ahead, Alan.
Right. So the oligos are fluorescently labeled. They diffuse really fast when they're by themselves because they're tiny. And when they bind to their complement on the viral RNA, they slow down a lot because now they're stuck to this big piece of viral RNA.
They basically move at the same speed as the RNA.
Right.
Yes.
So you're depending on this fluorescent property to be a readout for hybridization, essentially, right?
Right.
So in the old days, we used to use optical absorbance to measure hybridization because double-stranded versus single-stranded nucleic acids absorb at different ways. But you can't do that with the virus. There's just not enough stuff in there to give you a signal. So that's the power of this fluorescence correlation. So then the next step is to do the experiment. And what you can do here is you can take purified rhinoviruses and heat them at 56 degrees Celsius. See, this is why we use Celsius in the weather, because now everyone knows that 56 is really hot. Right? It's really hot. And it turns out that when you heat rhinoviruses and polioviruses at this temperature, they undergo a process that approximates uncoating. The first step is they become what's called an altered particle, which was the subject of Alan Dove's thesis.
Yep.
And this particle is conformationally changed and still has the RNA in it. And then they become empty particles. They're intact capsids, but the RNA is gone. And you can do this just by heating at 56 degrees.
The altered particle may or may not be the productive intermediate in there, but you do get this progression.
Right. Now, that was Alan's thesis. He concluded that, in fact, an altered particle is not and intermediate. And that bothered all of these people in the field like Jim Hogel and Dieter Blas, who have made a living. And to this day, they try and work around it in a variety of ways, which I never understood. But Alan does have a paper out there saying A particles are, how should we say, not intermediates, right?
Right. And it's been cited by like 50 different papers claiming that that's not the way it works. But anyway.
So they do their fluorescence correlation on virions that have been heated, right? And they add either one oligonucleotide for the 5' end or one for the 3' end. And they do their fluorescence correlation. And they have it expressed in a figure which I think is really nice. When they use the 3', they use it as percent of component. They take the data, the correlation data, and they express it in a different way so that you can see free oligonucleotide bound to free RNA, and then oligonucleotide bound to the virion via RNA. And you can see for the 3' end oligo, the concentration of the oligo goes down. The amount of oligo bound to virion initially goes up and then goes down, and then the amount of oligo bound to free RNA goes up later. This is within 15 minutes.
They're measuring this
or
over
so.
time.
Yeah, so it goes from 0 to 15 minutes. So the idea is that as you heat it, the RNA starts to come out, the 3' oligo binds, so you get this initial picture of the oligo binding to the virion, and then the RNA comes out, in which case the oligo is binding to the RNA in solution, which gives you a different pattern. And for the other oligo, the 5' oligo, all you see is it binding later on at 15 to 20 minutes as the RNA comes out of the capsid. Right. So that means that the 3' end comes out first, and the 5' end is later when the whole RNA is out. It's very cool.
I want to make a little aside here about on the topic I think I would call scholarship for the students out there listening, because I went through a process as I was reading this paper, because I first looked at these figures and saw these autocorrelation functions, and I thought, I can't understand this, and I cannot interpret the data. So then I looked into fluorescence correlation spectroscopy, and you look at some of the references, and after the obligatory couple of sentences that try and describe it in words, you get all kinds of math. And I said, I just have to understand this, and I spent time with it, enough time so that I finally got used to it, and I created my own sort of idea about how this works, so that by the time I got down to this figure 2 that you just described, I could look at the raw data on the autocorrelation function and understand what the heck went on. So my message is, don't be scared. Go ahead. You can figure this out. It is within your capability to understand this.
It will take time. That's the thing.
Yeah,
you can't be afraid of spending time on it, right?
Right.
Yeah, and I think we talked about this in the little bit that we did a while back on how to read a scientific paper, that when you look at these figures, these aren't splashy graphics for a magazine article that you glance at and it gets across one point. This is compressed data.
Right.
Mm-hmm.
And you, as scientists who are immersed in this stuff all the time, I mean, I'm reading papers all the time, and you guys are, and you look at something like this, we don't expect to be able to interpret this figure in 30 seconds.
Mm-hmm.
Right.
We're going to make we're going to make a day of this if we have to, digging through and say, what did they do here, and what does this graph mean, and at least piecing together enough pieces of it that we can make sense of what this is saying.
I've also learned to trust my own instincts in terms of asking questions, okay? When I look at something like this and I don't understand it, specific questions come to mind. And there's a tendency, one thing is to just say, oh, well, I can't understand it. I can't answer that question. But I think what's really helped me is to not let go of that. Follow that question. Just don't go read anything. Follow that question until you've satisfied yourself that you understand that question. And that's the way to learn about this stuff.
Yeah.
I had a slightly different approach, and that is that I really just tried to learn it from what they had in the paper. I knew if I had more time,
they do a pretty good job.
They do a really nice job, and I especially like this second part of Figure 2 where Vincent described the graphs of, that you can really tell what's going on. And so I think that's another lesson, that if the data are presented from a completely new approach, but in a clear and understandable way, the readers are going to be able to understand the point.
Yeah, it's really worth looking at Figure 2, and this, of course, is open access, so you can do that, because panels A and B are the correlation data, right? Mm-hmm. But it's hard to determine exactly what's going on there. You can see there's a difference, but when it's re-expressed as panels C and D, it's just brilliant.
Right.
Mm-hmm. You can see exactly what's going on.
By the way, the other approach is to ask your expert friend to explain this stuff to you, which sometimes works. And in this particular case, I couldn't even think of the expert friend.
Well, you don't have any friends, right?
That's true. That's a problem.
Are there any people at Gainesville that would know this?
I couldn't think of anybody right offhand. There probably are.
Yeah.
Okay. Probably. I'll bet you Rob McKenna can do this.
I would ask Steve Goff. He would know all this stuff. He would know everything.
And another thing that helps, I find, is to come up with an explanation for it as if you had to explain it to somebody else.
Absolutely. Yep.
And if you can do that, often I'll say, "Oh, okay. Now I'm going to try and imagine myself explaining this to somebody," and I very quickly realize, no, that doesn't float at all. I must have this wrong. Which is one of the tremendously educational things about doing TWiV. Yeah, actually.
Yeah, that's what we're doing here, right?
Snow in the headlights is brilliant.
Yeah. Thank you.
For those that maybe haven't seen that, I was thinking about dust in a beam of sunlight.
There you go.
Yeah, too. All right, so now they've got evidence that the 3' end comes out first. They want to confirm it by sequencing, see what RNA is left in the virion. And this actually takes a little bit to get to, but the first part of this is to use a chemical called sorolin, which, when added to nucleic acids, will cross-link them. So if nucleic acids are, say, double-stranded, it will cross-link them so they can't be denatured. And remember, one of the things that's happening here, when you heat the virions, you're altering them structurally, but you're probably also denaturing the RNA to a certain extent so it could get out. So they add sorolin to the virions, it can get inside, and it will cross-link randomly the RNA. And the idea is that then they heat these virions to 56, and as the RNA comes out, if a cross-link has occurred and left some double-stranded RNA, that'll stop the exit process. Right? So you can see then what's left in the virion. And if the 3' end comes out first, then mostly you should see loss of 3' sequences and not 5' sequences when you analyze what's left in the virion. Right, so that's what they do, and they treat their virions with sorolin, and then they heat them, and then they look at them in the EM, in the electron microscope. And there they see a variety of different kinds of particles. They have empty particles, they have full particles, and then they have particles that have some internal density within them that looks like a rod. They call it a rod-like appearance. Okay, so they interpret these that the exit of the RNA is halted at some point, and there's still some RNA in the virion, and that for some reason has assumed a rod-like shape.
So this is a constipated virus.
There we have a title: the constipated virus.
Go. Brilliant.
All right, so I remember the RNA is coming out of the pore.
Or snow in the headlights.
Snow in the headlights. Deer in the headlights, right? Then it reaches a sorolin cross-link, boom, you stop, and then the rest of the RNA stays inside. All right? Now, this is what I found really amazing. They say, to exclude the possibility that the rod-like density was a staining artifact, we did cryo-electron microscopy. Yes. which of course doesn't require stain. You just freeze the particles, and you take pictures of them, and then you do a lot of computational calculations and generate a 3-dimensional reconstruction.
Yeah, this struck me as quite an off-road trip to go on to eliminate an artifact, but it works.
Well, of course, having the structure is very informative, of course. So they do that. They do a lot of computational massaging here.
I showed this. I sent this paper to my structure buddies here at UF, and one of them, Rob McKenna, wrote back describing the structure in this case as a heroic effort.
Mm-hmm.
I wonder how long it took them to do that. Yeah. So there's a lot of terminology here which I frankly don't understand. I could probably figure out in a few weeks.
Right.
But the end
There was
result.
probably some interesting terminology when the PI first told the graduate student to do this
Yes,
experiment.
right. I mean, basically, they're able to solve the structures of these various particles that have, they're native, they have RNA in them, and then some with rods in them where they've been stuck. You can see the rod density inside, and they believe that from their calculations that the rod contacts a twofold axis. Now, this is inside the particle, right? It contacts on one side a twofold axis, and on the other side of the particle a threefold axis. So think that their idea is that this rod is what's left of the RNA in the capsid after it's been halted in its uncoating by the sorolin. And so the idea is it's either coming out at the two or the threefold axis because that's where the rod is pointing. And of course, they favor the twofold because it's bigger, but we don't know. You can't see any RNA outside the virion because it's probably just it's not ordered, so you don't see it in the reconstruction. Okay, so the rod inside is kind of an indicator of how the RNA is getting out. Okay, everybody okay so far?
Good.
Want to add anything to that?
Nope.
Okay. Now, they next do another kind of technology, which
Next, they go looking for the Higgs boson. No.
Wait, so we're moving off the cryo-EM at this
I'm
point?
going to move off. You want to say anything about the cryo-EM?
Well, just that because in cryo-EM you generate lots and lots of images, they have lots of them, and they could do. They separated these out into different categories of images, either that were full of the RNA or that had the rod-shaped thing in them, or empty. And that allowed, and they did finer and finer resolution. They started with 64 by 64 pixel analysis, and then they moved to 128 by 128. And I thought it was neat, and the figures where they actually show the cutaways with the rod inside of it, with the arrow pointing through it, and then how it rotates in the various views.
Right.
It's also worth taking a look at.
I'm surprised there aren't more dramatic differences in the structures, but I guess that was known. And not
that was known. If you look at these, you can see there are differences between the full and the empty, for example, and even the rod, the ones containing the rods which are on the way, are different as well. And that's been documented for rhino and polio before. Yeah. All right, so they use a technique called capillary electrophoresis to purify these different forms of the virus. So in other words, heated viruses with cross-link, and these, so you get these different states, full, rod-containing, and empty. You can actually purify them by capillary electrophoresis, and basically this is a very fine. It's actually a fluidic tube, one of these really tiny polymer tubes, through which you put very small volumes, and you apply an electric current, and you can purify different things as they come out one end. You put them in one end, you apply an electric current, they come out the other end. You can purify these three different types of virus particles, and they're able to do that. And they can look at them in gels and see the proteins that are present. So, for example, the full particles, of course, have all the virion proteins, VP1, 2, 3, 4. Alan Dove, what do the A particles contain? Which viral proteins?
They have lost VP4 and they've extruded part of VP1.
That's right. And then, of course, the empties also have lost the RNA. And so they can purify these partially extruded particles where the sorolin crosslink has stopped the extrusion. As you have a rod inside, right, and there's presumably a piece of RNA sticking out. And they use an antibody to double-stranded RNA, and they show that they can actually, that antibody will bind these particles.
Yeah.
Okay. I think this is cool. So the idea is that apparently this RNA, as it's coming out of the capsid, is refolding. These RNA genomes are all folded up, right? They're not just single strands. And apparently it comes out and refolds a bit, and it can be recognized by this monoclonal antibody.
The way I thought of that is that, they do the capillary electrophoresis on native particles, and they get a peak, and then they do it on the heated particles, and they get a new peak. And the presumption is that new peak is the stuff with the RNA sticking out of it, but they need to prove that.
Right.
And the way to prove that is to react it with the double-strand specific antibody and show that peak shifts.
Right.
Okay?
Mm-hmm. And also they can treat with micrococcal nuclease to digest whatever has come out on the outside and see the peak shift.
Yeah.
Right.
Right. So they take, in fact, these rod-containing particles. So again, these are particles, sorrel and cross-linked, 56 degrees for 10 minutes. They're frozen at some point in the extrusion. They treat them with micrococcal nuclease to remove what's on the outside, and then they amplify the nucleic acid that's in the capsid. So they extract it out, and they amplify it with oligos from the 5 at the 3' end, so they can see what's present in the virion and what's not. And they show that, in fact, the 3' end is missing from inside the virion, which means that it's extruded. Now, you may ask, why don't they just extract the whole thing without micrococcal nuclease? Well, of course, that wouldn't tell you anything because they just give you the whole genome, right? So the best you can do is to cut. I mean, it's kind of it's unfortunate that you have to lose the 3' end, but I think it's convincing because it's clearly not there. Right?
Well, what I like is they have two completely different approaches. One is the fluorescence correlation spectroscopy with the hybridization of the oligonucleotide to look at the 3' end, and the other is this purification of intermediates and a PCR method to look at the 5' end, and they're completely consistent with each other.
Right.
Yeah.
Mm-hmm.
Yeah.
Yes.
So they show that the 3' end is exposed to bind to the fluorescent oligo, and the 5' end is protected from nuclease.
Right.
So there you have it. So assuming that 56 degrees recapitulates what's going on in the cell, and it's probably close. I don't know if it's exactly the same, but the RNA comes out via the 3' end, and eventually the whole thing comes out, and it's oriented in the capsid in a certain way to allow this to happen. Right, so apparently, my guess is that the 3' end is right near the twofold axis, which is where it's going to come out. So how that happens is really interesting. It must happen during packaging, right?
Right. So, as I said, my first reaction was this is backwards, because if I were going to unpackage what is effectively a messenger RNA, I would do it 5' end first so that it can immediately interact with the ribosomes. But as Kathy points out, it may have more to do with the packaging, because as the genomes thing is going to be synthesized, the 5' end will emerge from the replication complex first, right?
That's right.
And so maybe that gets packaged first, leaving the 3' end when you're done in proximity to the channel. So does that make sense?
Yeah.
First in, last out.
Yeah.
Last in, first out.
Or last in, first out, if you prefer. Right.
Yeah. Right. That's another title: last in, first out.
And I think that if I understand the perspective correctly, another important part of this is reaffirmation that the twofold axis may be in fact where all the action is.
Yeah, they have. It could be 2 or 3-fold, I suppose. Right.
Right.
Because you can't tell because the rods are contacting both.
Right.
But I presume at some point we'll see some structure of extruded RNA, and that'll nail it. Mm-hmm. Right.
Yeah. And as I think they also point out that it makes sense that the virus would have a directional preference of some kind, if I can use that term, because if both ends came out, if either end could come out of the capsid, you could get a lot of situations where the virus has a chunk of RNA stuck in and both ends sticking out in different directions, and you might not be able to get uncoding.
Another interesting thing is that this rod, so in the native virus, that is the full virus, there's no rod, but then as it starts to encode it, apparently the RNA reorganizes to form this rod. So that's really interesting. Mm-hmm. I wonder what's controlling that and so forth. I think that's a neat question. And then the other I thought of, there must be some, certainly the replication, which puts the 5' end first, would result in this directional exit, but I wonder if there's some other purpose for having the 3' end come out first. There may be that we just don't know at this time, but
Right.
Interacting with some cell machinery that's important. These RNAs are thought to circularize when they're translated, so the 3' end, the poly-A binds poly-A binding protein. Maybe that is there and grabs it right away. Who knows?
Yeah, I just want to point out, I think they're pretty careful about saying that this is oriented with one end near a 2-fold axis and the other end near a 3-fold axis. They don't actually say it's at it.
Yeah.
Right.
Yeah, you can't. Yeah, I guess they haven't proven that, right?
Right.
It's just near.
And then the other thing that kind of blew my mind that, in fact, I had to write to Vincent about was the fact that when they want to get this sorolin into the particle, they just say at physiological temperature, small molecules can diffuse into the virus due to breathing. And that kind of, I guess it makes sense that, this metastable structure is already starting to destabilize a little bit at 37, but the fact that stuff can go in and dyes and sorolin and Vincent, you even said cesium for some of the picornis.
The rhinos,
Yeah.
And when you purify rhinoviruses on cesium chloride, cesium gets into the particle, so they band at a very different density than polioviruses, which exclude cesium. So, something shown by Roland Rucker many years ago. Yeah, it's pretty neat. All right, let us do a few emails here. Kathy, can you take the first one?
This is from Jeffrey. Illustrious doctors, there has been an idea floating around in my head since I read about rodents and bats in particular acting as reservoirs for numerous viruses. Perhaps their particular ecological niches put them in a genetic space where they are inundated with viruses and have had to evolve in that direction, but that could be said of many other genera which are not particularly noted as being reservoirs. This might be more common in these genera because, as with viruses, hyperadaptability is favored in species where reproductive rates allow for large numbers of throwaway, maladapted individuals. But I keep wondering if all of this adaptability and reservoir capacity might not also have evolved as a survival advantage in the form of defensive biological warfare. Isolated communities of a species adapt to the local virome and act as reservoirs for a number of viruses. Since the process of adaptation is somewhat random, each community and species develops unique reservoir capacities that even members of the same species over the next ridge won't match. As such, interactions with other species will always carry viral consequences. In the case of members of similar species entering a new community, they bring their own viral reservoirs with them, but in a kind of reverse herd immunity, their reservoir has less chance of overwhelming the native community than of the native community's reservoir overwhelming the intruder. Viral warfare defending territory and potentially leading to species isolation and divergence. In the case of, for instance, predator species, they generally have a lower reproductive rate and so are not as adaptable as their prey. Every time they consume a viral reservoir, they are at increased risk from that viral reservoir. In this case, the viral reservoir acts as a group defense, reducing predatory pressure. I strongly suspect that this sort of activity of viral reservoirs in rodents, bats, and the like exists on a statistical level, but the nagging idea in my head is that one or more groups might have evolved a genetic mechanism to enhance the defensive aspects of viral reservoirs. My questions for you experts are, is there any evidence that viral reservoir defense exceeds simple statistical effects? And how might you start to gather the data that you need to perform such an analysis? And how might you start to look for the genetic regulators of such a system? As always, thanks for the extra insights into the workings of this world. Jeffrey. I said that his question, how might you start to gather the data you need to perform such an analysis? I would start by asking somebody like Harmeet Malik or Sarah Sawyer or somebody who thinks about these things.
I sent it to Harmeet and he said he had to think about it.
I really, I just have to say, I really love this line
So
of
do I.
thinking.
Yes.
This is
That's
a
a very
really
brilliant idea.
cool idea.
It's a cool idea.
Even if it turns out not to be the case, it's I hadn't thought of this possibility, but wouldn't that be interesting?
So here's my problem. Evolution takes care of the moment, right? You have selection undergoing to make something fit for the moment. There's no long-term goal of current evolution, all right? So if, in fact, it turns out that, your own virome has some advantage, it's not something that happened by daily or hourly selection. It's just something that ended up being advantageous, okay?
But you would expect that if that did happen, if a particular population had an advantageous virome that prevented intruders from coming in, they would have an advantage over nearby groups that they might compete with.
Perhaps, right. But I don't think it evolved originally for that purpose. I think
I don't think things evolve for a purpose like that.
No, I mean, long-term stuff, evolution happens on a momentary basis and make you fit for the moment. So there's no long-term, we're not going to evolve for 10 years so that we could kill off this particular species. That doesn't happen.
Right, and speciation is not something that's specifically selected for. It's an artifact of the way the evolutionary process works.
Now, when I think of what kind of experiments you can do, I get really very nervous because I'm thinking about going outside and looking at communities and doing ecological studies. But I'll bet Harmit or Sarah can think of lab experiments that could test this, right?
Mm-hmm. So there's a couple of, first of all, there's a PLOS Pearls, which is a very easily readable little bit that we might want to link, talking about this notion that bats harbor a lot of viruses. So that's a nice summary of the situation. And in that article, they refer to a couple of bat genome analyses that have been done, and there's some relevance to what we're discussing in that, and I'll just read a bit from there. We would expect signatures of coevolution to be visible at the interface between bats and viruses, i.e. the innate immune system. Indeed, a number of genes involved in innate immunity were found to be under strong positive selection in the recently sequenced genomes of 2 different bats. Okay, so there's evidence from that point of view of bats adapting to harbor viruses, if that's the picture, over a period of time.
But, don't we think of, we think a lot about species barriers for viral infections, right? And your viruses are yours and not someone else's. So that could limit this. Mm-hmm. The advantage of unless you're talking about related species moving into a different niche and, then in that case, there are viruses, different species of bats, for example, or different species of rodents.
Right, and I think that's basically what he's talking about here. It's fascinating.
I think it's pretty cool.
I bet there's a lab system. Like, I know some people have done evolutionary studies on things like Daphnia, little water fleas. So there might be a system like that where you could see if you could induce some sort of separation in the viromes.
I know people have done ecological studies where, for example, to look at the role of sex in aquatic communities, you can look at the resident parasites and see how a non-sexual reproductive system compares to a sexually reproductive one. So in some organisms you have both, right? And you can see the effect of that on parasitic loads. People have gone out and done this in ponds, for example. So
right.
I guess you could do that. But we know so little about viromes out there in nature. That's hard, right? It has to be an experimental way to do this, a really clever way.
Yeah.
And I guess we're just not, we're just not clever.
Not clever.
We're not clever. Well,
it's a very cool question, though.
I like it very much. And so, listeners, what do you think? And maybe Harmit will give us some cool answers. Alan, can you take the next one?
Sure. Dave writes, "Hello, Mr. Racaniello. I would like to introduce you to a podcast I listened to recently through CFR. Here's the link. And this is the Council on Foreign Relations, I believe, not case fatality rate. And then here's a copy-paste from the CFR site. CFR Senior Fellow Laurie Garrett discusses pandemic preparedness on a global scale with professors and students as part of CFR's academic conference call series. It should raise your blood pressure."
I wonder why he said that.
Because Lori may say some things about how we're all going to die.
Yeah.
Because of high school students making terrible viruses in their garage.
Right. That's her shtick. Yeah.
That might raise your blood pressure.
She has said some rather inflammatory things about pandemics.
Yep. I'm not going to look at it because
I listened to the first 20 minutes of this, and she kind of more or less goes there.
I don't want my blood pressure raised.
There you go.
Yeah.
But I decided that everyone should know about it in case they have low blood pressure and they want to raise it. All right, Rich, you're next.
Robert writes, Dear TWiV hosts, I just wanted to say thank you for all the fantastic TWiV episodes all of you have done in 2013. I can't tell you how much my wife and I look forward to each new posting. I met my wife in my postdoc mentor's lab where she was a research associate. That was 38 years ago, and we are still academics at heart. TWiV feels very much like the lunchtime discussions we had with other students, postdocs, and faculty when we were at UCLA. We both look forward to another fantastic year of TWiV. Thank you very much. That's cool. The weather here in Orange County, California has been very mild. We did have a cold spell for a week or so where the day temps were never much above 60 degrees Fahrenheit, 15.5 C. The past week brought temps as high as 80 Fahrenheit, 26.7 C. I have been surfing and skiing since I was a boy, and I still enjoy both sports. He's really into the TWiV shtick here. This is good.
Yes.
Yesterday, along with some of my friends, we spent the morning at San Onofre, surfing in 2 to 3 foot swells and relatively warm water, 60 degrees Fahrenheit. We quit surfing around 11 a.m. and after driving home, rinsing out the wetsuits and eating a great meal prepared for us by our wives, we loaded up 2 cars with our skis and drove to Big Bear in the San Bernardino Mountains. We skied from 5 to 9 p.m., enjoyed another great meal, Mexican food, and we drove home. Southern California is a great place to live, I'll say. This is the life. Weather here today in Orange was 60 Fahrenheit, 15.5 C. Humidity 70%, dew point 49 Fahrenheit. Pressure 30.22 inches. I have a book I'd like to recommend. Quote, Life's Indispensable Molecule, end quote, by James Darnell, Cold Spring Harbor Laboratory Press, 2011. This book is a fantastic review that is accessible to anyone with an interest in the history of the rise of molecular biology and, in particular, the part that RNA has played. The author has spent 50 years studying RNA, and he has provided wonderful, well-referenced, readable story of those years. I also would like to recommend the book Biochemical Pathways, an Atlas of Biochemistry and Molecular Biology, by Gerhard Michal and Dietmar Schoenberg. Anyone who has ever been in a biochemistry laboratory will be familiar with the wall chart showing the biochemical pathways, which is currently in its 4th edition. This book presents the pathways, networks, and systems that have been elucidated with the advent of omics. The 2nd edition has been enlarged and contains the author's estimate at least 1/2 more material than the 1st edition. It is also available in Kindle. Once again, I hope you all have a wonderful holiday season and that the new year is filled with successful grant applications, accepted papers, happy and productive graduate and postdoc students, and TWiV episodes. Yours truly, Robert.
Nice.
Nice
story. I think you can surf and ski in the same day. Cool.
Yeah, amazing, huh?
Wow.
I'm guessing maybe Robert is retired now, or maybe this was a weekend.
I don't think so. I think he might be. Yeah.
He's having a pretty good time.
38 years ago, and we're still academics at heart. So it sounds like he might.
He might be retired.
Which sounds pretty good. And then you get to listen to TWiV. Hey.
Still surfing and skiing. That's
nice.
Good.
Thank you, Robert. Very nice.
I don't think I would buy a biochemical pathways printed book, though, because it's got to be out of date by the time the ink is dry.
It said it's available in a Kindle.
Yeah.
Right.
I never saw this RNA Life's Indispensable Molecule. Anybody read that?
Mm-mm.
No? It's 2011, brand new. Check it out. All right. The next one I'm going to save for Dixon because it's up his alley. And let me write that here so I remember not to print it. Yeah. You know what I really like? We all work on a Google Doc as we're doing TWiV, and you can see who's doing things. Because when someone makes a little edit, their name is appearing above the cursor. It's so cool in real time. Let's see. The next one is from David. Hi. I'm an MSc graduate of virology and currently working as a research assistant at Urmia University of Medical Science, West Azerbaijan, Iran. I want to thank you and TWiV for all these great podcasts, but sadly I should tell you that we did not know about the TWiV and its podcast until recently, and since then I recommend it to all my friends in the field of virology in our country. I have searched among all episodes but did not find any TWiV about the human cytomegalovirus and its reactivation and latency. If it is possible, please make an episode about it in future. Thanks in advance. I don't think we did a CMV, right?
No, we've discussed CMV on numerous occasions, but we've never done a CMV specific episode to do the whole thing.
Well, at ASM, we had Tom Schenk talking about CMV.
But we didn't talk about reactivation and latency,
No,
I guess. He
that's
talked
true.
About metabolomics. So, yeah, I think that would be a good topic. I will say that in 2014, we'll do one.
You know, early on in the history of TWiV, you did try to do virus families kind of one at a time over a period of time. And I know that because you had me on to discuss pox viruses.
Yeah, that's right.
But that sort of concept has gone away over time.
Yeah, that was early on when I was sort of, trying to see what would be a good thing to do. And
we might want to revisit some of that.
Yeah, we could certainly get someone who does that and talk. I think it's great to do it now and then for sure.
Yeah.
Iran,
so that one that episode where Tom Schenk was on was 234 live in Denver.
That's right.
Okay.
And I just when I saw this letter, I had to Google West Azerbaijan because I thought
So
Azerbaijan
did I.
I thought, wait, that's a country,
Yeah.
but there is a province in Iran called West Azerbaijan, and I'm sure there are people who are very upset about that. But anyway, it's
Well, I think it's cool that we have listeners in Iran.
yeah.
I didn't know that there was much virology there, so cool. To all you virologists, TWiV fans in Iran, hello.
Yep.
Let's do just one more here from Robin. This is he writes Pleo and Poly. Remember we had an email about why we use
Pleomorphic and polymorphic.
Yeah.
Right.
So he says Pleo is a continuum of variation and poly is multiple but discrete. Did you guys know that?
Nope.
Now I do.
Thank you, Robin. Always
Robin keeps us on our
keeps
toes.
us on our
Yes.
toes. All right.
Pleo and Polly sounds like it could be the name for a cartoon or something.
You could name your cats that.
It can.
Yes.
Just what I was thinking.
Actually, I want to just move one up here, which is a cartoon, because I think we should.
Yes.
This one's sent by Victor. Thought you might appreciate this, and this is a Peanuts comic. What's the guy's name?
Linus.
Linus says,
Linus.
why do I have to get a measles shot? Whoever worries about measles, what's a little rubiola among friends? And Lucy says, your stupidity is appalling. And Linus says, most stupidity is. Let me move that up in the queue here so we'll get it. That's great.
Yes.
Let's see. Paste. There we go. Thank you, Victor. We do appreciate that, right?
Yep.
Yes.
So I have to remember that whenever we say, why are these students? Well, you get it. Never mind. Let's do some picks. I scroll down here. Scrolling. Got a lot of emails here.
A lot of good emails. They're really great.
Yes.
Yeah, we have many more. But keep sending them in. We'll get to them. Okay, Alan, you're the first
Okay. My pick is a YouTube video, so careful if you click it, it'll autoplay. This is a time-lapse video shot from the cab of a locomotive on what's called the juice train. And this is it's a freight train. It's one of these little glimpses into the industrial infrastructure that hardly anybody ever notices. But 5 times a week, this massive freight train full of cases and cases of orange juice goes from Florida to New Jersey. And they shot this video from the locomotive cab. It's
I'm getting dizzy.
Yeah, GE put this thing together because they built the locomotive, and they're hyping, how fuel efficient it is. But which is good. But the video itself, I thought, was really cool, and it follows the train along on its 48 hours straight shot up the East Coast from Florida to a freight yard in New Jersey, and then the orange juice gets distributed from there. So you walk into the grocery store, you see the orange juice is always sitting there, and you never really give a thought to how it got there. And
along with everything else.
Along with everything else, exactly.
They make a few stops, I guess, to change engineers and stuff, right?
Yeah, they have to change shifts, and I don't know if they have to refuel.
I have to say, now I am proud to live in New Jersey.
There you go.
Because that's where the orange juice goes.
That's where the orange juice goes.
I'm proud to live in Florida because that's where the orange juice comes from.
Yeah, you get all the big bucks.
Yeah, but there could be problems with that bacterial invasion, right?
Yeah, that's always a problem. That big microbes and the citrus industry are a big deal down here.
Yep.
Yeah, which is why freezing is a big deal down there, too.
Yeah, you don't want the oranges to freeze. Rich, what do you have?
Alan just segued into my pick, which was sent to me by my good friend Grant McFadden, which is a space shot of what the most recent storm looked like going through the Great Lakes. Whiteout over Great Lakes seen from space. And this is a really nice image. That's actually if you look at the details, it's a bit of a composite of a couple of different images. It's got visual and infrared overlaid on a standard satellite map so you can see what's going on. But it shows the Great Lakes just basically whited out because of this storm. Grant is from Edmonton, Alberta, and spent many years in Western Ontario, so he has experienced this. And you may recall that I lived for 12 years in Buffalo, so I paid my dues as well. Lake effect storm is where the Canadians send cold air down across the Great Lakes from the northwest, where the prevailing wind is from the northwest, and it flows across the lakes, which are still warm early in the winter, and picks up the moisture on the lakes and then freezes it and dumps it wherever it is downwind, and that just happens to be Buffalo and a bunch of other cities downwind of the Great Lakes. And this is a wonderful image of what that looks like.
Yep.
So,
yeah, look at Western Michigan.
Yeah, it looks awful.
And they always get so much more snow than we do, or almost always. But yeah, there I am. I'm waving. Can you see me?
Yeah, for sure. Alan,
I guess this was also that polar vortex, right?
Yep.
Right.
Alan, you know the author? She graduated from the NYU program in science journalism. Gianna Briner.
Gianna Briner. That sounds really familiar.
Yeah, she's down. You got a little bio at the
This
bottom.
actually is a pretty cool site, this Live Science. I kind of looked around a little bit. I couldn't quite figure out how it was put together, but it's a nice website.
Kathy, what do you have?
Okay, I have to get off of that map and go back to mine, which is also a maps pick. And it's funny because I had just heard this piece of information on another podcast I listened to called Away with Words, that, when kids are out digging and they're digging a hole to China, unless they're in parts of South America, it's not going to happen.
Yeah.
They're going to hit the Indian Ocean. Yeah.
Yeah. This map shows in green areas that are where there's also land on the other side of the world. So take the bottom of South America and then look over there in China, and so you have to be in South America to be able to dig to China.
Okay, so if you don't want to drown, you got to dig in one of the green areas to go through.
Right.
That's cool. That's very cool.
Right.
So nowhere in the U.S. can I get to land on the other side.
Apparently not.
Well, there's that little thing that I can't tell whether it's in Canada or the U.S. A tiny little dot.
Ah, yeah.
And if you go up from that dot, you can see the green landmass, and then that's part of the Antarctic, and it's some island there between Africa and Australia.
Huh.
But you don't know where you go if you go in these green areas, right?
Well, you can kind of figure it out.
You can
look
match
at the
them
shape.
up.
You can map it, yeah.
So look
like
at
if you look
Spain
at the easiest
New
is the southern
Zealand.
tip of South America.
Yeah.
So where do you go if you that little spot in Canada, northern U.S. Canada? Where would you
Go
wind
to
up
that
at some sort of island.
Yeah,
little
that little
dot,
island that's
between
south of,
Africa and Australia. See
Yeah, I
that
see.
tiny
Oh, I see.
Yeah, again, look at New Zealand and Spain.
Got it.
So if you dig a hole straight down
okay,
from
yeah.
up there in Canada in the middle of nowhere, you come out in the middle of nowhere.
Oh, the shape tells you where you would come up, right? Because
Yeah.
there's New
So-
Zealand and Spain. Yeah, got it.
So it's a little bit like seeing the rod in the icosahedron.
Yeah,
On at the twofold axis and coming out at the threefold axis. You
There
have
you
to
go.
be able to visualize it on a flat thing, but think in 3 dimensions.
right.
It is interesting that from I guess parts of Spain, you would end up in New Zealand.
That's what I think. Yeah.
Yeah.
Nice.
And if you want to dig to China, you got to start in South America.
Yes.
That's cool. You have to give that to kids so they can dig to the right place now.
Yeah.
That's China.
Exactly.
Okay, I have a interesting little artsy pick. I was on Twitter earlier this week, and I saw a tweet from Michelle Banks, who's a microbial artist, and she said, "Oh, these necklaces, they definitely look like viruses." So I went on over to this website, which is a website over at Etsy called Loriola, and it is by the artist Lori something. What's her name? Lori Flanders. So she makes glass, and she makes various things with them. And one of the things she makes is necklaces. And I'm telling you, these components of the necklace look like virus particles.
Yep.
And they're beautiful. They're just gorgeous. They're all different colors. So I actually was chatting with her over on Facebook, and I asked her if she was inspired by viruses, but apparently she just likes to make beautiful things. And I think it's pretty neat that she ends up making what look like virus particles. It just shows that viruses are beautiful.
Yeah.
Well, she's got one of these that's called infectious.
Yeah, so I don't know. That wasn't up when I originally emailed her or Facebooked her. And then she said that she was going to put more up that night, and maybe she called it that because we were talking about it. I don't know. Maybe she knew it.
Cool.
Right.
But anyway, there are 4 or 5 of them that...
Yeah, so you guys are killing me because last week Alan's pick was the Wubble, so I bought one of those. Vincent sent me this link earlier in the week, and she had a sale which I didn't quite know about, and they cost differently depending on how many of these viruses there are per necklace. But anyway, I have one of those on order now too. So Rich, you have to see if you can pick something next week that I'll want to buy.
Okay. So there's probably, a Porsche or a BMW that can fit this.
So these are cool. These are handmade glass.
Yeah,
she puts them
they're
together,
beautiful.
and then she puts them into necklaces. And it'd be interesting to see how big... It's hard to tell actually how big each virus particle is.
Well, on one of them, if you click on the arrow to the right, it showed on a mannequin.
Yeah.
Right.
They're substantial.
Yeah. Yeah.
Well, when you get it, you'll have to wear it for us on TWiV.
Yeah. Yeah.
Right, that's cool. We had 2 listener picks, one from Carol, her knit picornavirus, and 2 books of Roberts. So that'll be our listener picks for today. And you can find this episode of TWiV at iTunes and twiv.tv. And do send us your questions and comments to twiv@twiv.tv. And if you have a moment, go on over to iTunes and rate the show with a couple of stars or a comment that helps to keep us visible so people can find us. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun, and I really did learn a lot this week with these papers.
Yep,
It was fun.
those papers are cool. Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And Rich Condit is back at the University of Florida in warm Gainesville. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.